Okay, go for it.
Welcome to talk number 2, everybody. My name is Chris Trevetti, and I am a graduate student working in John's Computer Lab, which should tell you a lot about me. And how crazy I am. My work involves studying microbial communities found at a cold water sulfidic spring Canadian high Arctic, more specifically looking at how those microbes have a role in sulfur cycling and biomineralization. But I think John's going to talk about that a little bit more later today. So what we're really here for is our next speaker, Dr. Laura Baumgartner. And Dr. Baumgartner received her bachelor's and master's degrees from the University of Colorado Boulder in environmental, population, and organismic biology. She went on to the University of Connecticut, focusing on biological oceanography for her PhD. After that, she joined the PACE Lab as a postdoc and lecturer. And nowadays, she is at Front Range Community College, where recently she was named the Master Instructor of Science, which is a pretty big deal. Dr. Baumgartner also has a decade of higher education teaching experience and 2 decades of outreach teaching. So without further ado, let's welcome her today to the Normposium.
Thanks, Chris. And I realized after hearing Dan's speech, I should have said a couple things, Chris, because I was at University of Connecticut, but I was actually mostly being taught by one of Tom Smith's former students, Dan Buckley. And when Dan abandoned me for Cornell, I was actually fortunate enough that John and Norm actually housed me for the last 2 years of my PhD. And that was key to most of the progress of my sequencing in my PhD. So I have to put appreciation there. And as a statement of John's training, I do have to include a where we're going slide, because John always says you have to tell them where you're going and you got to tell them where you've been. So hopefully I'll remember to tell you where we've been. So I have actually 2 things to talk to you guys about today. One is the microbiology of the built environment, or the Sloan project as it was known in the lab because it was funded by the Alfred P. Sloan Foundation. A little bit about the evolution of that project, where it came from, and some research examples from the project. And then my life in this talk will take a sharp left turn, because I actually went into education. And education has changed a lot from what it was like when most of us were in the classroom. Talk a little bit about where those reforms came from, and a little bit about what my classroom actually looks like. And I do have to make one correction. I misinformed Chris, because the master teacher was actually for all the campus, and it was the first time science has won in 20 years. So myself and my mentor actually won the 2 prizes. So to start, Sloan program. Sloan program got its start with Paula Zalowski from the Sloan Foundation, and she asked some really cogent questions a lot of people hadn't been asking. And a lot of these came from a program that came about after the anthrax scares of the early 2000s. And BioWatch program was a set of filters that were set up in several cities across the US, eventually 30 cities, sampling for various bioterrorism agents. And very quickly what they found, particularly in the Southwest, was that they got positive hits that were not indicative of bioterrorism events. And this was kind of surprising, except to some people it wouldn't have been. So when we talked to the vets, the hits were for anthrax and tularemia in particular. And when we talked to the vets, they said any place that's been near a cow field probably has a load of Bacillus anthracis. This isn't a shock. But it was a shock to a lot of people, because it turns out we had no idea what baseline looked like. We didn't know what normal air was. So Paula Zalowski from the Sloan Foundation asked these questions. Nobody was really asking, what's normal? What is it we're being exposed to in our everyday environment? What does the built environment look like? And what does the outdoor air look like? So she started this research program and approached a few key researchers, including Norm. Norm she approached partially on the basis of the shower curtain biofilm study and the therapy pool study that Norm talked about last night. And pretty quickly we realized part of the reason a lot of these answers weren't known is nobody quite knew how to sample. So this slide is actually from a meeting that we held with one of the companies that we were trying to buy air samplers from. And as Norm mentioned last night, fortunately we had collaboration with the Hernandez lab, because the company was not so surprised that we wanted samplers that were quiet or that had a high sampling flow rate, but they were really surprised by these bottom three. Their standard format include vibrating plastic together, which created these little tiny bits of plastic that fluoresced under an epifluorescent microscope. Couldn't do a DAPI count to save your life with that. They were really shocked that we wanted to be able to clean this out in the field and take a second sterile sample. They were only set up for, oh my gosh, you've exposed it to bioterrorism agent, and now you would like to clean it with super caustic chemicals. So that was kind of a shock. Hernandez's lab had been working with several different samplers, and over here on the left, the liquid impinger was pretty much a gold standard at the time, but it had a flow rate of 12 liters per minute. It took 30 minutes to an hour to get a decent sample, at which time you replace the sample liquid in it several times, which makes sterility and everything else a little bit difficult. The one over on your right, the BioCapture 250, had a really high sampling rate, but look at the efficiencies. If you're interested in the fungus, it was pretty much useless, and actually in my hands, it shot all the sampling liquid out the front end for some reason, so the two of us never got along. But there were several samplers that Hernandez lab and our lab worked through, and we finally ended up with the Scepter Omni 3000. 270 liters per minute. It takes about 10 minutes to get a cubic meter of air. That looks pretty good. So we bought one, and then we realized we had a different problem. Normal air sample over here on the left, DAPI, there's not a ton in there. And then over on the right, that's what our samples look like. And anybody who's used to looking at stuff under the scope, there's not a lot of diversity in there. That was the first thing that made me nervous, and then I realized the counts were really weird. The entire machine was an excellent, excellent growth chamber. And they didn't have a great way to take it apart. So, look at the bottom of this thing. They had come up with this really nice mechanism to replace the liquid that you lost as you were sampling, because you wanted it to evaporate. It took me an hour and a half to take that mechanism apart and strip it out to be a new machine after every sample. So after 3 generations, we got this thing over on the right. It took us 10 minutes in the field. And we could verify it to get a completely clean sample. So later when Mark Hernandez asked me to teach his microbiology for engineers class, I was a little bit motivated to go into that first class and explain microbiology to engineers. While we were fighting with that, Mari Rodriguez de Eptifoff and Jeff Walker went into New Orleans with one of the older style samplers between Hurricanes Katrina and Rita, and then back again after Rita. And there's kind of a hypothesis. You have a city that's completely flooded with water. You would expect that to somehow affect the air that you're seeing, particularly when they went down there and started to see that they were moving the water with these huge, basically sprinklers. They were aerosolizing all this water that had been sitting over the city. And in reality, what they found was the shared microbiota between the air and the water is fairly minimal, which is kind of interesting. It was one of our first signs we didn't quite know what we were looking for. Norm actually spoke about the showerhead biofilms. One of the things I find interesting about this, which is much like mine being housed in the lab for a couple of years, very indicative of the PACE lab, this study actually started with Norm and I helping a methods class at MCDB. And I have to point out what the students really wanted to study, and this is a study that still needs to happen, was beer taps, and why dirty beer taps give you a headache. So if anybody's still looking for research. Norm had to KO that one, and I suppose now that I'm teaching, I understand why. Norm's talked about this study, but what he didn't talk about was how that got received in the press. We listened to the Psycho theme song for a long time after that one. The mycobacteria that we found in the showerheads kind of just kept coming back. Kristen Peterson did a study of not therapy pools, but actually recreational pools, and again found mycobacteria really like that environment. Lifeguard lung is known in both environments. Norm talked about the drinking water study. There's the mycobacteria again, so that's Eric Hollinger and Kim Ross. And Julianne, and I'm trying to find I'm stopping because I need to acknowledge, and I told him I would forget, Kirk Harris, Dan Frank, Mark Stevens, and Chuck Robertson, we noticed on almost all of these papers. But Julianne and Kim were the main ones for this one. Hospital water, with and without chloramine treatment. And this is kind of the first one of a theme that also shows up how the water's treated, and it shows up in the previous study as well. How the water's treated seems to be the bigger theme. So not your geography, not all the things that we thought would matter as you went down the Arkansas, but how you treated the water creates some big changes in what you actually see. I found the magic button Dan found earlier. This is one study that I always wanted to play with, and Natalie finally did it. The humidifier question. So what the heck, when you put a humidifier in your house, what is it you're spraying out into the air? So again, those little questions every day, what are we exposed to? And the Sloan Project has covered a lot of them. So leaving the water side, this is the stuff we had to think hard about, was the air. And the subway study was my personal baby. And I have to say, only living on the East Coast for 4 years set me up for the joy of watching Norm, and Kim's smiling, watching Norm walk through New York City with a ski bag over his shoulder. Because nothing got as many second glances from the New Yorkers as that. The ski bag actually held the stand for the sampler. And the other part was, and that's Paul Ozlowski actually talking to you there, but the New Yorkers would all pop off the subway cars, take one look at us, and then intentionally look away and walk as fast as they could. No one ever walked up. I'm so used to being asked what I was doing in fieldwork. No one ever walked up except once, right after the explosion at Grand Central Station. We were sampling at Grand Central. This woman walks up to ask what we're doing, but it wasn't the, excuse me, can I ask what's going on? No, it was a New Yorker. What the hell is that? So, at least I was ready for the East Coast again. Subway study came with its own interesting problems, which, again, if we asked the right people, all the subway workers knew this. There's a huge load of iron in the air of the subway systems, and after working all day in it, we knew it too. So the two filters on the left are subway samples, the other filter is an outdoor air sample. And you can see the brown on the filters. And fortunately for me, it turned out to be magnetic iron, so I could remove it just by exposure to a magnet, but did add the complexity of making sure that we did, in fact, not disrupt our samples after that. And Norm talked about the subway data yesterday. So there's a lot of stuff that's not known. There's a lot of stuff that resembles the environment, which if you think about the open grates in a subway, makes sense. And then there's a lot of stuff that looks kind of like us. And Norm didn't point out my favorite part of this, because you start with the things, the plantar heel. Norm talked about the bellows walking through the city. Start with the things that are fairly exposed, and then you get to the stuff that you try to think isn't actually exposed when you're wearing clothes. So that always amused me. Chuck did a fabulous analysis on that one. Norm gave this picture last night. Switch over to the other indoor air, and I was laughing when Norm puts this picture up, because this is the other part of that picture. See the sampler way at the very top? We were 30 floors up, and it's the catwalk at the top of this hotel. And this was the view down in the corners. And this is how I found out that neither Perrette nor I were all that fond of heights. That trip was also interesting because we got sampled inside an HVAC system. I don't know how I got volunteered for that one, Perrette. Sitting inside the HVAC system for 10 minutes with the sampler. So that was, you thought field sites and indoor were going to be a little bit less interesting, but you never know. So Perrette was the leading force on the studies of shelters, healthcare. We did schools. In Atlanta we covered, and in the Boulder area we covered hotels, office buildings, all sorts of other buildings that are in there. And what we saw more than any other place, even the subways, is indoor air looks a whole lot like humans, which is what Norm was talking about last night. We did find that engineers have actually done studies showing that there's that 37-degree plume coming off of us. We're influencing the air that flows in the environment, in our environment, and we're certainly influencing the microbiota. This one gets the award for most interesting grant to write. I don't know if it was Norm or Dan Frank who came up with the phrase manure splatter, but getting that into a grant was a skill. Contained animal feeding operations, the main lead on that was actually Mario Rodriguez de Eperhoff. And we studied swine barns, which unfortunately Mari has the pictures from because that was amusing, and cattle barns. And the main thing that came out of this between Mari and Perrette was the grant was from NIOSH, and one of the things we were looking at was worker exposure. And not surprisingly, what you're exposed to changes as you enclose the barn more. So swine barns range from fairly open, big open sides that look like garage doors basically, to very enclosed, and you can see complete enclosure really changes the microbiota that the workers are being exposed to. Not a lot of signs of pathogens still. The other part of the study actually looked at circovirus, which is a known swine pathogen. Generally, when a farmer knows they have circovirus, they will take out their entire herd. And what Pret found was that every single barn seems to have circovirus that we looked at. So it's not the thing that comes occasionally, it's the thing that's there, and it's whether or not they're actually seeing symptoms.
Can I inject
Yes.
more? This is an arena where this aerosol sampling is really, use a lot of development. It's a lot of money talking. Virus detection, what we show is that we can detect viruses with aerosols. Now, in general, a public health type person who would go into a facility looking for things, they'll walk around and do some swabs. You may or may not get something. But with aerosol, you're getting a comprehensive view of the entire facility, and their use is not being used in the industry. And particularly as we begin to worry increasingly about not only disease coming in from animals, but intercontinental, even transported disease, with water communication. How do you monitor this? This will, it's cheap, it's very efficient, it's very effective. And I don't know if anybody's developing this.
I don't. But Pret can speak to how hard it was to do it the first time. I think now that she's got it figured out, it's pretty doable.
That's pretty for swine flu and stuff like that.
Can you get access though? Do the people running these want to know?
Well, I think there is an issue, but as the regulatory aspect, people right now, our disease problems in some of the worst populations we've done. And enough said, it's an industry that developed.
They're open to the extension services coming in, and that's how we snuck in. Because, yes, it took a while to get context. Last study I want to point out from this side is actually Allie Ling's, and she was actually looking at how the microbes affect the environment we build. She was on Mark Hernandez's students mainly, but again, one of the many that Norm housed and helped. She was looking at corrosion of concrete and the diversity of the microbes that go with it. And what you can see is she's looking at time-wise as corrosion progresses, diversity compared to everything else you're seeing goes way down. And you got a pretty good lead on what sort of organisms are associated with that, which leaves pretty good suggestions to how we can fight that corrosion. So the built environment has become quite the field. This today is actually up on MicroNet, but Sloan program has built this huge set of studies. When we started, there was barely any research to lean back on, and now if you go to MicroNet, they list the centers that they're funding. My favorite thing is they regularly post recent built environment microbiology papers. June 3rd edition has 5 papers. They post bi-monthly. We had almost nothing to lean on, and now there's 10 papers a month in the field. That's a huge change, and what we're learning is pretty amazing. And here's where I'm going to take a sharp change. This is not a Google image search, I suggest, although it comes very close to one of the slides. Norm always talked about the fire in the gut. You always need to follow the fire in your gut. And much like Norm probably realized things about Dan before Dan did, Norm realized things about me before I did. And I liked research, but it wasn't my fire. I loved teaching. So Norm pushed me to do more and more of it, and he was very supportive when I switched sides. And teaching has changed, like I said, since most of us were in the classroom. Some of that change is linked to Eric Mazura, who in the 1990s was teaching at Harvard. He's a physicist. And he came up with this idea called peer interaction, because he watched all the students, and he stood there and did a standard lecture and talked and talked and talked. And realized the students weren't learning a bloody thing listening to him talk, except what it was he wanted them to put on the test. But they weren't connecting it with their daily lives. And I know this is not news for some of you, there's other people in the room that are on the forefront of this stuff. But he came up with this idea of you do the part of the talking you want to do, and then you ask the students what they just learned. And then he did the thing that was really inventive. Not just asking them what they think they just learned, but asking them to talk to each other about it. And that has hugely changed how we teach now. A lot of studies have actually been done at CU in the MCDB department, and I was fortunate that Norm let me spend a lot of time with Jenny Knight and Bill Wood who've looked at this stuff. And what they have in the top graph is after they ask a question to students, what percent were correct. Second question is after the students have discussed it. And then the third bar is a similar question, just phrased differently enough the students aren't going to recognize the answer immediately, that might be given the same day, might be given 2 weeks later. And what they found, particularly as you'll see for the more difficult questions, is that letting students discuss with each other helped them get to the right answer. Never did they actually give a definitive right answer on any of this. And the crazy thing is, nobody in the group that's discussing needed to actually have the right answer when they started. So if you follow this line over here, 48%, most of the group was incorrect. Those are the groups that are listed as 48% incorrect. Nobody in that group had the right answer to begin with. Second question, half of those people, half of that 48%, so about 25% of your students can answer the second question right. And then from that, 77% can answer the third question right. So, and if you start with your other group, 90% of your first group was answering it all right at the beginning. They quickly figured out, you don't need them to know, you need them to talk about it and think through it. And I enjoyed a lot of collaboration in the PACE lab, and it kind of set me up to collaborate outside of my field once I started full-time teaching. And one of the philosophy teachers that I work with has this thing, you need a mission statement. What do you want to achieve? Beyond your standard learning objective, what is it you want to do? And I want my students to think and analyze. And in pure lecture format, if I just talk and they write as fast as they can because I can talk fast, how much of that are they doing? So I kind of had to wonder, what do they want? What do I want them to know ahead of time? Because if I want them to talk to each other about the subject, they have to have some clue about the subject. And then what can I do change in class? And one of the biggest changes I made, sort of like when none of us dared go to lab meeting without having some clue what was going on, is making them do their work, starting with the textbook. And every person here who's taught knows the most common phrases, but they won't read it ahead of time. They won't. I have online quizzes. They have to have read it ahead of time. And I keep a little humor in there. My favorite YouTube that I lean on is Crash Course, and one of theirs is water. So intro, science class, how does water work, the 4 traits of water. And one of his statements is, the things that benefited from surface tension are several insects, 1 lizard, and 1 Jesus. And there's nothing quite like opening a quiz where all my students answer this last question, 1 Jesus. It's like, how much trouble am I going to get in for that one? In the classroom, there's a lot of resources that didn't exist, certainly didn't exist when I was TAing with Norm. Case studies, simulations, and those of you who looked at PhET once a long time ago, they've switched from all physics to including a lot of good chemistry, and they're slowly adding biology. Slowly. It's getting there. My slides got out over there. So then there's 1,000 tools that you can use in class. All these point to asking students to rephrase what I just told them in their own words. Can they say it again? And I don't use clicker questions because I'm not at a big school where it's required. I actually use whiteboards because they're awesome, because they can draw me a molecule, they can show me a pathway, or they can agree to have their photo used in a conference they won't be at. It's always handy that way. Biology theater. This is the biggest thing. Students are incredibly willing to be cheesy and act out photosynthesis. You can be an electron transport chain. It turns out they love it, which took me a while. There's different models available. If students can tell you what something is and isn't, that's great. If they can make the connections, that's great. I can't do a concept map to save my life, so I always have to use the fill-in ones. Lessons all the way across. When we started Sloan, when I started teaching, the first thought you always want to go to is something super elaborate. And all the researchers in this room can tell you the simple study that gets you the simple answer and it's clear is often the better one, and Norm often had to remind me of that. Collaboration. PACE Lab is an amazing place for collaboration. Norm has always been willing to bring in people that weren't even his responsibility and collaborate with them. I'm learning to collaborate with students. I actually let them teach several of my classes at this point, which is awesome for me, I have to say. All the way around, and this is something Norm taught me about research and has worked in the classroom too, be willing to play. You want the answer to that, just go do it. So, huge thanks to Norm. Cheryl and John for organizing and, quite frankly, for keeping me sane for several years. Paul Islowski and the Sloan Foundation for funding. And I want to re-acknowledge the influence of Dan, Kirk, and Jeff, and Chuck on the Sloan project as it started. And then most of the others are people who were part of the Sloan project at some point. So thank you all. And I will say several of the other people who've done the research, some of it was after me, and I did make one bobble there, are in the audience. So if you have questions, I may field them outward, just to warn those of you who are sitting here. So thank you. Yes.
I get great things like charades. And I do it all the time, and I love it, but I don't know how to grade.
I give 1 point a week for being there and being active. So a lot of it goes into that. Bigger things I actually have rubrics for, and I make everyone fill out the rubric for everyone else.
Yeah.
And students are amazingly harsh. So it's useful. That's everyone else super happy.
How do you I was particularly struck by the Psycho cartoon, and how kind of blending your teaching aspect into research side. How do you avoid the public being scared of the findings that showerheads have?
I actually have been starting my class now with that study. And I actually have my students read a newspaper article that was published about the showerhead study, by finding pretty much the scariest one I can. Oh, your showerhead's trying to kill you, et cetera, et cetera. And then actually look at the study and compare it. And I spent a lot of time on how does what you see in the news actually reflect the science, because it is a very small piece of a much bigger study, and people, so much is open access now, you can usually go back and find the study and figure out what they were actually trying to say. So I do spend a lot of time on that with students. I would encourage anyone else to do the same.
Can I follow up on that just for a second? So, when the shower curtain study came out, remember when Rush Limbaugh picked it up on the radio? And Rush Limbaugh's like, there's this guy out in Boulder, Colorado who has all his time on his hands to look at what's on a shower curtain. and that kind of built momentum. At some point, I think you were in Vogue too, right?
I was trying to find a picture with Norm's name in Cosmo with the guy in the shower.
Right.
I couldn't find it.
What becomes critical of that is really like, it builds on its own. You release this thing, and then it grows and goes. You can't control that. I think good science writers can kind of help that, because they put accuracy out there first, and other people draw from those first stories that come out, and that kind of gets the message going in a good direction. And I think that's where good science writing plays a role in trying to get accurate information out. That's just a thought I have.
There's a practical public health issue showerhead story, and it ties in with the municipal water story, the high levels of mycobacteria, some of which are known not good things. And this is the nature of the showerhead. As soon as we started this, or I didn't mention it, but when we had showerheads from around the country, because her hubby was an airline pilot, and so she thought about swab out showerheads, sample.
He's really good at sampling if you need internet, cross-country samples now.
The thing that impressed me is, given the amount of microbes that are slushing around in our regular environment, the issue becomes not, is the pathogen present? We know they are, the opportunistic pathogen. We know they're there, and the question is, how are they supplied? And if you're taking a shower, which generates a lot of aerosol, steam, it's not steam, but it's scalding, it's aerosol. Shoots right into the deep air rooms. I'll bet that that's the source of much pulmonary mycobacterial infections, which is endemic and epidemic in some parts of this country. The statistics for non-tuberculous pulmonary disease are very poor. Docs aren't looking for it, but even with diagnosed, as it is, the incidence, the prevalence in this country is 100 to 150 cases per 100,000. In some parts of the country, it's 300,000. In some places, in Hawaii, it's over 400,000. That's it. And the epidemiologists start talking about epidemics of 150, 200 people. So here we have this poor diagnosed disease all over the people. Bottom line, shower. You want to use a shower as opposed to taking a bath. Get a low aerosol shower. stay away from these new high efficiency showers. But I can take a shower, leave it in water. It's delivered, it's a 35 minutes. Yeah, shoes, everybody's.
We advised everyone at the time to get metal showers, but can you even get them anymore?
I just want to say that I was just at the American Thoracic Society meeting in Denver a couple weeks ago, and it's an international gathering of 15,000 pulmonologists. And one of the keynote sessions was to shower and not to shower. And it was a debate on the stage in a very large auditorium between Rachel Thompson and Kevin Penley. Rachel was the pro-showerer, and Kevin was taking the non-showering. And I can say Rachel won that debate in my query of the audience. So it still goes on, it's raging, this debate.
The question is the nature of the shower. It's delivery, you're going to get a rain type shower in probably. It's okay compared to this shower.
And those fine details didn't come out.
Just imagine what would happen if we done beer taps instead. I'd still drink.
Let's take a break till 10 a.m. There's coffee and tea in the atrium. Bathrooms are around the building.
Just gonna get my
Hold it.
No.